The organization of life. Cells are the building blocks of living organisms. They are the smallest, organized, self-contained living systems. They can be entire organisms by themselves or only one of billions of cells making up a complex human being. We all came from the same starting point and, despite the long and convoluted path of evolution, have retained many features of our tiny one-celled ancestors. By some measures, 5% to 7% of our DNA is the same as that in bacteria. We truly are connected to all other living things on Earth. Cells are tiny bags full of organic compounds, but just as those compounds are intricately organized chains of carbon and its atomic buddies, cells have their own structure and key bits. Broadly, there are two types of cells in living things, prokaryotes and eukaryotes. Prokaryotic cells carry their genetic material in a region that is not enclosed by a membrane like the nucleus in a eukaryotic cell. Eukaryotic cells protect their genetic material with a special double membrane wall that surrounds the nucleus of the cell. Prokaryotes are mostly bacteria. Why wouldn't bacteria want to protect their DNA? Prokaryotes are thought to be the first living cells to evolve, so they're phylogenetically older than eukaryotes. The membrane surrounding the nucleus evolved later. And they've also done pretty well without nuclei. They are very well adapted to harsh environments. We don't fully understand why prokaryotes are so adaptable, but their hardiness and adaptability have made them the most abundant life form on Earth. Bacterial cells are typically much smaller than eukaryotic cells and have a variety of shapes. They are highly successful in part because they can reproduce rapidly when the circumstances are right. Under the best conditions, some species can replicate a whole new generation in less than 20 minutes. This rapid rate of replication gives bacteria the opportunity to profit quickly from random mutations. With short generation times and large populations, even rare new mutations can give the species an advantage when it comes to surviving new environments, because they have more opportunities to develop mutations that might protect them from the elements. This is one of the ways in which bacteria can quickly develop a resistance to antibiotic medicines. Another quality that enables antibiotic resistance is the unique mechanisms by which prokaryotes transfer genetic material, or DNA. They can take up genetic material from their surroundings and incorporate it into their own code even when the new genes come from a different species. Despite the fact that 1% to 3% of our body mass is bacteria, the rest of our bodies consists of eukaryotic animal cells. Remember that all cells need to sustain and regulate themselves, reproduce, and help the organism grow. All of these functions require cells to be highly organized. The cell's membrane, or cell wall, is made up of lipids, so it's water-fearing, or hydrophobic. This is so that the contents of the cell don't leak out. However, if the entire membrane were hydrophobic, then the outside of the cell would repel water that carries nutrients to the cell, like oxygen and glucose. The cell needs these nutrients to survive. Structurally, lipids resemble jellyfish. They have a spherical head attached to some chains that bring to mind long tentacles. The spherical head is hydrophilic, or water-loving. The tentacles are hydrophobic. The cell membrane has two layers of lipids organized so that the spherical heads are on the outside and the tentacles meet on the inside. This organization allows the spherical heads that are water-loving to be in contact with the water. Scattered throughout the membrane are proteins that act like gates. They help some desirable molecules get across the cell wall and keep the unwanted ones out. Once we get past the cell wall, the next most obvious part of the cell is the nucleus, which also contains a membrane in eukaryotes. Just like the outer cell wall, the nuclear membrane has a lipid structure, but for added security, the membrane is doubled. That is, there are two membranes, each with a lipid bilayer. The wall also has pores, or holes, that facilitate the passage of certain large proteins and macromolecules. The main job of the nucleus is to house and protect the cell's genetic information, which is stored in the form of DNA. Proteins are what make a cell function and display the properties of life, and to make proteins, the cell needs to use the DNA blueprint to manufacture and assemble these complex molecules. Inside the nucleus, the all-important job of using the DNA code to generate messenger RNA occurs the first step in the creation of new proteins. Each gene along a DNA molecule permits the synthesis of a specific mRNA, which then interacts with the cell's protein factories to direct the assembly of proteins and their parts. In the nucleus, the DNA creates mRNA, which is then sent out to the protein factories via the pores in the membrane surrounding the nucleus. These protein factories are called ribosomes, and they come in two types, free and bound. Free ribosomes float around in the cell while bound ribosomes are attached to the endoplasmic reticulum, a network of sacs and tubes that serve as the industrial zone for the cell's various large manufacturing projects, such as the synthesis of new membranes or protein gates. All of these different components of the cell underscore the very fundamental characteristics of life: sustenance, reproduction, regulation, and even adaptation. There are three more key parts to the cell: the Golgi apparatus, the cell's storage and shipping warehouse, the mitochondria, the energy generator, and the lysosomes, the waste management system. In organisms as complex as humans, there are many different types of cells, from neurons to pancreatic cells to blood cells, and each one has additions or subtractions that support its specific function. The ways that those different cell types interact took hundreds of millions of years to evolve. But the cell parts discussed are common to most, if not all, cells and serve as the building blocks for our bodies. There is some debate as to whether a virus should be considered a living or a non-living thing. A virus cannot replicate or power itself, it has no metabolism or ability to generate energy from its environment. It depends on host cells to make new proteins. However, a virus is made up of the molecules of life, DNA, so it can replicate only with the help of a host and, therefore, evolve. These features make classifying viruses as living or non-living difficult. The complexity of single-celled organisms is remarkable, but in some ways, even more remarkable is the fact that billions of seemingly self-sufficient cells can work together to form complex beings like humans. If we really want to understand why one of the fundamental properties of life is organization, we need only to peek inside the most complex thing we've discovered so far in our universe, our brain. Our brain is made up of about 100 billion cells, or neurons. Each of these neurons is connected to other neurons, and information flows from one cell to the others. Some neurons get their information from as many as 10,000 other cells and send it on to thousands more. The signals that are sent between neurons are binary, just like the code that our computers use. They either encourage the downstream neuron to send its own signal or to keep quiet. However, if we look more closely at the junction between one neuron and the next, the story quickly becomes far more complicated than that. The space that separates one neuron from another is called a synapse. More specifically, a synapse is the spot where one neuron sends its information and another receives it, like a dock in a shipyard. Ships come in with containers full of different products. Many of those containers are accepted at the dock, and they release their contents into trucks or other vehicles that then transport them to other locations. Some containers can get lost during the transportation, some are rejected by customs. Some ships don't even drop off cargo, they just deliver instructions for the customs officials to use. At the synapse, many neurons dock their axons, the part of the cell that sends out electrical and chemical signals. The receiving neuron, or the destination, also has many docks, or dendrites, to receive the signals. Scattered along the dendrites are customs gates, which analyze the structure of the molecules that are released by the axons of the upstream neurons. Some of those molecules are accepted, and the signal gets propagated down the dock and into the core of the receiving cell. Some are rejected and never make it past the synapse. And some are lost during the journey, they never make it to the dock. All of these different molecules and their interactions have an effect on the functioning of the cell and its downstream connections. Some molecules are involved in making memories, and they change the way that the neurons get connected. Some turn on or off other functions, like those that cause the cell to fire off a signal or stop it from firing. Then, if we move a level up in terms of the organization of the brain, we find that there is a pattern to the electrical signals that neurons generate. Certainly, these signals are affected by neurotransmitters, the molecules that travel across the synapse, but there is also a code in the firing patterns that can be just as important in terms of making memories or relieving pain. For example, cells send signals by altering the rates at which they fire, or by firing in bursts or in synchrony with one another. Each of these codes can be related to different brain functions and malfunctions. The brain's intricate organization can also be seen in the neuroanatomy of different brain regions and the connections between them. For example, the hippocampus's complex neural architecture is responsible for creating memories. Thank you for listening them.